Assalamu alaikum wa rahmatullahi wa barakatuh. Welcome all, and thank you for joining us. In 2017, the FDA approved the first CAR T-cell therapy for acute lymphoblastic leukemia and certain lymphomas, ushering in a new class of drugs based on T-cell engineering. Our keynote speaker today is the 2025 King Faisal Prize in Medicine Laureate, a globally recognized pioneer in chimeric antigen receptor T-cell immunotherapy, a groundbreaking approach to cancer treatment. He led the development of CAR T-cell targeting CD19 and established genetic engineering and cell manufacturing capabilities to translate this research, which enabled him and his team to start treating patients with refractory leukemias in 2007. His lab obtained the first FDA approval for a CD19 CAR T-cell therapy trial in the US and was the first to report dramatic responses to CD19 CAR T-cell therapy in adults with relapsed and refractory acute lymphoblastic leukemias, leading to long-term survival. Professor Michel Sadelain started with a baccalaureate degree in math from France in 1976, then went on to obtain his MD degree in 1984 from the University of Paris Pierre and Marie Curie, followed by residency in internal medicine and clinical immunology. Soon after, he completed his PhD in immunology from the University of Alberta in 1989, followed by postdoctoral fellowship at MIT. In 1994, he joined Memorial Sloan Kettering Institute and was appointed as an assistant professor at Cornell University Medical College. Later, in 1997, he was appointed director of the Gene Transfer and Somatic Cell Engineering Facility, and in 2004, he was appointed professor of immunology at Weill Cornell Medical College. Later, in 2007, he founded the Center for Cell Engineering at Memorial Sloan Kettering Cancer Center. In 2024, Professor Sadelain joined Columbia University as the inaugural director of the Columbia Initiative in Cell Engineering and Therapy from Memorial Sloan Kettering, where he was the director of the Center for Cell Engineering. Has been elected to several medical and scientific societies, including, among others, American Society for Clinical Investigation, American Association for Cancer Research. He has authored over 280 papers, with his research leading to the creation of over 60 patents. He served on many NIH committees and several scientific and editorial boards. He has mentored and supervised over 80 graduate students and postdoctoral researchers. Among his honors was receiving the Breakthrough Prize in Life Sciences in 2024, and on this coming Monday, he will be awarded with the 2025 King Faisal Prize in Medicine. So please welcome with me our esteemed keynote speaker, Professor Michel Sadelain.
Well, thank you for this very kind and long introduction, but thank you all for the invitation and for being here on a weekend, I know. And thank you for your very warm hospitality. Talking to some of you already over lunch, I noticed that many of you here had trained at Mayo Clinic, Vanderbilt, MIT, UCL. I feel like I'm in New York, actually. So I'm going to tell you about CAR T cells, how they came to be, and focus on some of our current research in this lecture. And I'm going to start from the very beginning, from evolution. And evolution has given us an amazing immune system that protects us against a wide range of pathogens, viruses, bacteria, parasites, and obviously if we didn't have an immune system, our species would not be on this planet. And over time it has evolved many layers, many systems within this immune system, culminating with what we call adaptive immunity, which are sets of immune cells, namely T cells and B cells, that can acquire highly specific molecules known as the T cell receptor or antibodies, again, to protect us against these pathogens. Now, T cells in particular are formed in the thymus, which is where the letter T comes from, and they acquire through genetic recombination what we call the T cell receptor. It's a blind recombination of genes. By blind, I mean it is not targeted to any particular antigen. The thymus selects T cells that are potentially useful to us, as had predicted actually McFarlane Burnett, shown there in the picture, way before all of this had been discovered. And all of this gives us what we call a repertoire, which is a collection of different T cells that we have in our body, which is estimated to comprise about 20 million different TCRs, T cell receptors, in a normal adult. And that should protect us against, most pathogens perhaps that we might encounter in a lifetime. Having said that, this repertoire, as vast as it is, it's not infinite. And perhaps evolution didn't have in mind cancer. Cancer is not an external invader, but rather arises from the acquisition of somatic mutations in our own cells. And sometimes we do have T cells that recognize our cancers, but often we don't. As you all know, cancer most of the time wins the battle. This graphic that you see here is one that certainly all the oncologists will be familiar with. We modified it here, but it shows you the accumulation, the frequency, if you like, of mutations in different cancers. And some of them circled, do I have a pointer maybe? If there's no pointer, do you see my arrow? Oh yes, okay. So I circled here in gray these cancers that have a high number of mutations. 100, sometimes 1,000 or more mutations in cancer cells. And these mutations change the sequence of normal proteins, and some of which might become immunogenic. And for some of those, one might have a T cell receptor that is specific. And in fact, those that are circled in gray here are the cancers that tend to respond to checkpoint blockade immunotherapy, precisely because one might have specificities in the immune system for those antigens. But you see all the cancers circled here in red. A lot of these have a low mutational burden. And so the probability that you would have a T cell capable of recognizing one of these mutated exons known as neoantigens is far less. Which brings us to really the reason for the rationale, if you like, for wanting to instruct the immune system to recognize antigens of our choosing, and to do so with a different receptor, not the natural receptor. I'll introduce CARs in the next few slides, but I'll just start by saying that CARs don't recognize antigens in the same way as the T cell receptor, which, again, the immunologists will know, engage HLA complexes that present peptides processed inside those cells. No, CARs recognize molecules on the surface of cells. But those could be peptides, it could be proteins, it could be a carbohydrate, it could be a glycolipid, as long as they're on the surface of a cell. So they're not HLA restricted. Furthermore, and this is relevant to cancer, one of the mechanisms that cancers adopt to escape from an immune response is to downregulate HLA. If there's no HLA on a tumor, there can be no antigen presented, and then no T cell receptor could engage them. But if your receptor is independent of HLA, you can engage, again, HLA negative tumors. The third argument for CAR T cells is one I already stated, is this low mutational burden. If there's a low content in neoantigens, well, you could look for different kinds of antigens. And lastly here, the rapid response, really, that's not a feature of CAR T cells alone, it's a feature of any adoptive cell therapy, which aims to provide cells to a patient, bypassing the need for immunization or vaccines, cells that are, ready to go. So it's hard to summarize, the many years that brought this therapy, but I will highlight here 4 critical moments, if you like, or milestones in the development of CD19 CAR therapy, which I'll explain. The first one is really establishing methods to introduce genes into T cells. And so, yes, I was, for myself anyway, in Canada, and at the time, working on my PhD in immunology, and I wanted to teach T cells what to do, what we want those T cells to do. And it was clear that that instruction would have to come in a genetic form, and for that you would have to figure out how to introduce genes. And so I went to MIT at the time because gene transfer technology was barely starting, hadn't been applied to human cells yet, it was barely applied to mouse cells. Anyway, we established methods for the efficient introduction of genes into T cells. The next milestone really is the creation of these receptors that guide the T cell to the cancer. And one way of telling this story is to start from the cloning of this, what's shown in green here, this green chain, which is the zeta chain of the CD3 complex. The CD3 complex mediates T cell activation. And 3 groups, I have nothing to do with this work, in 1991 cloned that particular zeta chain and created these fusions that are shown here. They created fusions because these zeta chains don't have an extracellular domain, and they didn't know what these zeta chains did, so they created the fusions, introduced them into T cell leukemias with antibodies. They could cross-link the extracellular domain and realize that the zeta chain initiates T cell activation. And then 2 groups in 1993 altered these fusions by placing here as the extracellular domain the single chain fraction variable, which are the heavy and light chains of immunoglobulins on these zeta chains. One of these 2 groups is Zeli Geshar, who was in Israel. He called it a T-body. And in doing so, they could redirect hybridomas to recognize an antigen of the choosing. But because we had recently established means to introduce genes in T cells, we realized that these chains didn't work, or didn't work well. They would allow a cell to kill, and then the T cell itself would die. And of course, with that, you would never have a therapy. So we started studying these various molecules here. I call them CCR, chimeric co-stimulatory molecules. Anyway, that can, if you like, fuel the energy in T cells. And eventually we understood that we could create a molecule that would target T cells to recognize a tumor antigen, allow those T cells to kill those targets, but then most importantly, allow those T cells to further expand and to continue to function, which would be critical to establish a therapy, by fusing different signaling domains, in particular here CD28 and CD3 zeta. And while today that seems to be obvious because there are so many of these molecules, it was an idea that sort of broke a taboo at the time of merging these signaling domains into a single receptor. These are the molecules that we called CAR, and introduced this terminology of chimeric antigen receptor. Now, to develop this, we needed a target. The target would have to be a cell surface molecule. And we thought that if someday we were going to bring this to the clinic, it perhaps should be a hematological malignancy, because, for lymphomas, leukemias, well, obviously there was an unmet medical need, but also sort of a tradition of doing bone marrow transplants and maybe a greater acceptance, if you like, by clinicians to introduce T cells into patients. So we're looking for a molecule and chose CD19, which is present in a majority of lymphomas, leukemias, was known already to be there, and in normal B cells, and I'll come back to that. It was also, we favored it over better known molecules such as CD20, in particular because it's more abundant. And that turns out to be very important, as I will say later. And so in this paper, to call it a landmark paper by now, in 2003, we showed that you could take human peripheral blood T cells collected from a patient or a healthy donor, introduce into those T cells a gene using a recombinant retroviral vector coding for a synthetic receptor that we call the CAR that was specific for CD19, and that a single infusion of these T cells given intravenously to tumor-bearing mice would result in these long-term remissions and even cures. And when that study was published, we and some others realized that this really should go to the clinic, but there was no industry interested in this. So if you were going to develop it, you would have to do it yourself. And at my center, we decided to do so, and I recruited Isabelle Rivière, shown here, also from MIT, who established this laboratory, GMP laboratory, if you like, that enabled us to do the first trials. And I'll come back to what two other centers did. And so these are the first two molecules that were approved by the US FDA in 2017. These are chimeric antigen receptors that recognize through these external domains CD19. They have this dual signaling structure that I already mentioned, one for activation and a second component that increases the strength of signaling and really enables the T cell to continue its work, if you like. The one on the left is the one I already mentioned before. It's the first one. Uses this costimulatory domain board from CD28, and the one on the right uses the costimulatory domain borrowed, if I can say, from a molecule called 41BB. That was first published by Dario Campana at St. Jude. And so we brought this to a clinical trial, and two other groups in the US also brought this to a clinical trial. The National Cancer Institute, to whom I gave our sequence. Steve Rosenberg used our sequence. And then Carl June at the University of Pennsylvania, he got the car from this Dario Campana that I mentioned, and he conducted trials too. It was a good thing in the end that three centers, we would have liked to be the only ones, but it was a good thing that three centers tested this because each one of them reported very early, unexpectedly dramatic results, which are complete remissions in patients with relapsed refractory leukemia or lymphoma. So the NCI first reported on one patient with lymphoma who received one of these CD19 CAR T cells and went into complete remission. The University of Pennsylvania was the first to report on three patients with chronic lymphocytic leukemia, and we were the first in acute lymphoblastic leukemia, and that's the example I just highlight here. And so we reported on the first five patients, and you see here, these are CD19 positive cells here in the bone marrow just a few days before CAR T cell infusions. There's all these blasts that you see in these patients with relapsed disease. And you can see within days here on day 11, for example, the complete obliteration of any CD19 positive cell. And so this was in the news and in many places, and these results were soon to be confirmed in larger studies. Science proclaimed that year cancer immunotherapy to be the scientific breakthrough of the year, and that was largely for checkpoint blockade, these antibodies that awaken the immune system in some tumors and many tumors, although not all patients and not all tumors. And they also highlighted what was just starting, which was these CAR T cells, and later in trials, and here's a review, these results were really confirmed in larger studies, so they weren't just arising in small studies. And it's when I was explaining this this is our local newspaper, trying to explain to a journalist how this works. You take cells from the patients, there's no drug, no, the medicine is a cell. It's a cell that is genetically reprogrammed. It uses a synthetic receptor. You need a modified virus to introduce this gene into the cell. It sounded very complicated. And I just told her, well, look, we're creating living drugs. And that's what this T cell is. It is alive and multiplies, but it's been instructed by this genetic modification to perform the task that we would like it to perform. So there are, as of today, 7 CAR T cells that are approved in the United States. 5 of them target CD19. They use either one of those two signaling domains, and 2 of them target BCMA, B cell maturation antigen, and that is an antigen that's relevant to multiple myeloma. And here below, I just give you some of these complete remission rates now reported in large studies. This is, so-called real-world data in different B-cell malignancies or multiple myeloma. Again, all of which are obtained in patients that have refractory relapsed disease, right? These are patients who have exhausted the standard of care, often relapsing or after chemotherapy and sometimes a bone marrow transplant. And so you can see in certain lymphomas, the complete remission rate is very high. 80% in acute lymphoblastic leukemia is very high. And even in diffuse large B-cell lymphoma, which is harder to treat, these complete remission rates, again, are very remarkable for patients who don't have any other option. Now, I could give a whole lecture on the toxicities of these molecules, and I don't have the time today, but I thought I would still mention it. Of course, all therapies have some toxicities, and there are 4 of them that come with CAR T cells. The first is a cytokine release syndrome. You know, some patients, when they get these T cells, the T cells get very active in sort of a synchronized way, and there's in some patients an excess of cytokines. We didn't see this in mouse models, but we saw this very soon in the first clinical trials. It was a surprise in the beginning, but today this is very, or almost all the time, easily controlled. And we published a few years ago a mouse model, which took a long time to come up with, that taught us about other target molecules to mitigate this cytokine response. The second is a neurotoxicity. In some patients that receive these immunotherapies, and by the way, not just CAR T cell therapies, there are neurotoxicities. You know, it can be brain fog, sometimes more. That pathology is not very well understood. We know there can be damage to the blood-brain barrier. It doesn't require the transfer of T cells into the central nervous system. Again, the pathology is not very clear. The third one is a small fraction of patients will develop neutropenias or thrombocytopenias. They're reversible, but a small fraction, they're durable, and again, the mechanism is unclear. It's thought that there is some damage to hematopoietic stem cells in some of these patients. And the fourth one is rare but dramatic. It's maybe 1 in 2,000-3,000 patients, so it's really quite rare. But on occasion, some lymphomas have been observed. There's a few reports up to last month, including one of ours, but it's still unclear whether the CAR plays a role in these lymphomas, because T-cell lymphomas, because a number of these patients also who received prior chemotherapies and bone marrow transplants sometimes do develop a lymphoma, and so it's not very clear if the CAR T-cell is contributing to that or not. Anyway, I did want to mention these. What I really want to discuss is where we go from here. And I liken what's happening to this field now to the Cambrian explosion, which for those of you who will recall your zoology, is some very long ago, maybe 250 million years ago, when there were all these life forms that appeared on our planet. So this was a formidable time of diversification of species on this planet. And to some degrees, the chimeric molecules that we're creating today, starting with CARs, are a little bit shown here on these left, all these exotic chimeras. Another explosion of molecules, of human-made molecules that recombine natural components, but to provide new properties to human cells. And so I'm just going to illustrate some directions in our research that today contribute, I guess, to this Cambrian explosion. And I want to discuss the antigen sensitivity of CARs. So the CAR T cell is there to recognize a cancer cell, and it recognizes cell surface molecules. The question is, how many molecules are needed on the surface of this tumor cell for the CAR T cell to effectively find the tumor cell, to stop there, bind to it, and then go on to kill it? Like all things, there's a limit and there's a threshold, and until recently, it wasn't known at all. So, then I'll discuss something that's a little fancy here in the new designs of CAR T cells, where the term of logic gating has been introduced, and I'll explain what that means. And then I'll address quickly also 2 aspects of persistence of T cells. Persistence refers to how long can the T cell act in the body, and obviously this living drug has to live long enough to fight the cancer. And not just survive, but survive in a functional state. If not, it's not a medicine. So I'll start with the antigen sensitivity of CARs. And there are 2 ways that we've addressed this issue. First of all, we wanted to know, how do these CD19 CARs actually recognize the antigen? And it wasn't known, so I teamed up with investigators at Cornell University who did cryo-EM to look at how the two first SCFVs that were used in the clinic actually recognize CD19. This is the SCFV that I consider the original one, the first one that we used, but then everyone else in the field used FMC63, which is another SCFV, because it was available in the public domain. Interestingly, you'll see that these two SCFVs almost bind exactly the same site on CD19. Anyway, with that information of their binding, we could, with the help of computers, predict, an energy to each amino acid residue within these SCFVs and rapidly create mutants, single point mutations in these SCFVs. And I'm just going to show you an example very quickly here of what we can do. This, you can see, was published. So here we start from one of these SCFVs that is introduced in a CAR, and with T cells, we're killing in vitro tumor cells that have different levels of CD19. This one is very typical, has about 20,000 CD19 molecules on their surface. And then we create these variants that have, you see, 2,000 or, for example, 200 molecules of CD19. And so I'll just take one of these mutants, and it doesn't matter which one it is, it's the orange one. You can see in vitro, it kills well when there's 20,000. When there's 2,000, it starts to not kill very well, and when there's just 200, it fails completely. So you can design SCFVs with different affinities that result in different sensitivities of these CAR T cells, and this is one technique to more rapidly get what you're looking for. But the bigger issue in the clinic is the escape of tumors that don't have enough antigen to be recognized. And this, again, was brought to our attention by clinical results. And here I show data that's not from my lab, it's from a paper in Stanford that nicely reported on this. These are, you see here, 3 patients with diffuse large B-cell lymphoma. And you can see that the amount of CD19 on their surface is quite different, and actually lower than the amount that you see in normal B cells. And we and others then started noticing that some patients would relapse after CAR T cell therapy, and with the antigen, the targeted antigen still present on the tumor, but at a lower level than prior to treatment, suggesting that the CAR T cell, for some reason, couldn't recognize those tumors. Here's a very clear example with CARs targeting CD22. You see, this is the number of molecules on these leukemias and from these patients before treatment. And when they relapse, yes, some are negative, but a lot of them are lower, and they tend to be lower than before. So we went back to this T cell receptor that I mentioned before, the natural, the physiological T cell receptor, for which there's a lot of data showing that it is highly sensitive. It can recognize fewer than 5 HLA peptide molecules on a surface of a cell. And it signals through this CD3 complex. You recognize that green chain here I showed in the beginning. On the far right here are the CARs that I'm talking about, which with their dual signaling domain and recognizing antigen through heavy and light chains. We wanted to compare the CAR and a T cell receptor. But to do that comparison, we wanted the T cell receptor and the CAR to recognize the same thing. So we created this structure, which is shown here, where we swap in these heavy and light chains, replacing the so-called variable alpha and beta chains of a T cell receptor. This is done through genome engineering in peripheral blood T cells using CRISPR-Cas9 and incoming donor DNA for homologous recombination. And we call this receptor HIT, which stands for HLA-independent T cell receptor. It recognizes here the same CD19, for example, as this CAR. Anyway, this work was published long ago in 2022, and we showed that indeed a structure such as this one is a lot more sensitive than the CARs that we traditionally have used till now. I'll say more about this in this other lecture at the end of the day. I'll show you an example, though, of what we can do with these HIT receptors. This is unpublished work now, where the target is no longer CD19, but CD70, and the disease is renal cell carcinoma. so cancer in the kidney. And we're targeting PDX models, right? Patient-derived xenografts. And so here we place the PDX in the lungs of these mice, and here in the kidney. And then we treat them with CAR T cells, targeting CD70. And you can see that in the lung, there's a very beautiful response. These tumors are nicely eliminated. But our CAR T cells fail. They fail in the kidney. Even if we give very high doses here, we never give doses of 10 million CAR T cells to a mouse, they still fail. And there could be many reasons for that, perhaps T cells not entering the tumor, etc. But we soon realized that it's about the target. And here's an immunohistochemistry of this PDX model in these mice. And you can see some of the cells stained positive for CD70, and a lot appear to be negative. I didn't mention, but in culture, these PDX models are completely CD70 positive. And here, if you remove the PDX from these mice and examine the level of expression of CD70, you see here in the lung, there's a lot of CD70, and in the kidney, there's not. You see that. It is the same PDX in the same mouse, okay? And this is another PDX model, K7. Again, yes, CD70 is abundant in the lung, but it's downregulated or apparently negative in the kidney. But we soon found out that although they appear to be negative, and they're negative by immunohistochemistry, but as you know, that's not very sensitive, they actually do express CD70, but at a very low level. We could show that by proteomics, by RNA, etc. And so the question for us was, well, would our HIT receptor now work? And that is the case. That's what's shown here. So again, here's the kidney model. In blue and purple here are mice treated with the classic CARs, if you like. But if we engineer these with the HIT receptor, you see the green lines here. There's a very precipitous decline in tumor burden. There's relapse in these mice because these T cells don't persist so long, and if we confer additional properties to increase their persistence, you now see in red this very rapid decline. So the HIT receptor is highly sensitive in vitro and against a range of tumors, even those that might appear to be negative on immunohistochemistry. And we're studying this mechanism now. It's about epigenetic regulation of the target. I won't go into this, but you see if we sort out these CD70 low kidney cells, their expression is low. Just in culture, it comes back over time, coming back to the level that you see in those cells that are positive, or in the lung, or in cultured cells. And we now recognize that it's about methylation of some histones that is different in different anatomical locations. And so we're pursuing this further to better understand what controls expression of this target. And so the point I wanted to make, though, today is that the field is starting to have a set of different chimeric antigen receptors with different levels of sensitivities. And some of these, like the 4-1BB CARs used in many instances, they require at least 1,000 or 2,000 molecules on the surface to be effective. Those that use the CD28 CAR are a little bit more sensitive. They can still work if there's as few as perhaps 500 molecules on a tumor cell. And then with the HIT receptor, you can, at least in mouse models, since this has not yet gone to humans, certainly the animal models show you that you can target as few as 200 or 100 molecules per cell. So I want to discuss now another aspect of this. We call it logic gating of T cells. And here we go deeper into, you could call it bioengineering or synthetic immunology. The T cells normally have one T cell receptor. Every T cell recognizes one antigen. But if we're engineering cells, why should we limit ourselves to that? Why not have T cells that recognize 2 or even more antigens? Now, if you just take the case of 2 antigens, there are actually many ways of doing that. And the field has now adopted this terminology of algebra, or Boolean algebra, to describe these gates. So the most common and most obvious gate is, you could call an OR gate. This is a T cell that would have 2 receptors. It can recognize A or B. And this is a commonly used gate. Obviously, you can diversify what your T cell can engage. Another gate is called an AND gate. This would be a T cell that would only recognize A and B together, but not a normal cell that has A alone or B alone. And you can guess this is because it's not always easy to find tumor-specific targets, but maybe there are combinations such as AB here that would be unique, as long as normal cells express A but not B or B and not A. It's not an easy gate to come up with. We showed one 10 years ago. Recently another group came up with a slightly better one. It's an interesting gate, but not, these are diagrams, not so easy to do. A NOT gate comprises an inhibitory signal. So here's a CAR that recognizes antigen A, and here is an inhibitory CAR specific to antigen B. And when we describe these, we first used PD-1 based inhibitory CARs. PD-1 can inhibit T cell responses. And so the idea here is that you have a CAR that could recognize antigen A, but then antigen A may be on some normal cells, which would result in toxicity to those normal cells. But perhaps you can find an antigen B that is not on the tumor, but on the normal cell, and therefore protect the tissue with an inhibitory receptor. The fourth gate is now called an if-then gate. This introduces a temporal dimension. You see here a receptor that engages antigen A, and the best known system was described by someone called Wendell Lim at UCSF. It's a Notch receptor. It engages antigen A, and this leads to the transcription of a CAR specific for antigen B. The idea there was to restrict expression of the CAR to the tumor microenvironment so that the CAR would not be constitutively expressed. And then the last gate is what we call an if-better gate, and I will explain in a minute what we mean by that. But simply said, this is, you see a CAR here engaging A. Maybe there's lots of A on the surface, so the CAR T cell works well. But if there's a limited amount of A, the CAR wouldn't work anymore. So can we help it? And provide a second molecule, a helper molecule, if you like, and that would engage antigen B. So the CAR anti-A would work better if antigen B is there. So first I'll say a few words about the OR gates. And this one we described in a project for AML. And we're combining two CARs here. One is a HIT receptor, which is sensitive, and we're combining with a CAR, one of those more classic CARs, although not the conventional one. It's one called 1XX that I'll present later. The issue here is we want to effectively target two antigens, so one cell that really does a good job against two different antigens. And the way we test that in the mouse models is by creating tumors where we purposefully mix tumors. So this is an AML, an acute myeloid leukemia, that expresses the two antigens are AGRE2 and CD70. But we created these variants by genetic engineering, where this has AGRE2 but not CD70, or this has CD70 but not AGRE2. So if your T cell works well, it has to kill all three. Because if you only kill two of these, your mouse will still die by progression of the fraction that is not killed. So we tried different combinations of CARs, putting two CARs in the T cell. Those CD28 CARs, these BB CARs, recognizing this one or that one. And we could not get a good combination, but we finally, whoops, how do I go back? Maybe, oh, I don't know how to go back here. No, oh yes. And you see when we create this HIT and CAR, thank you, then we could finally get a combination of these that work well. You see here survival to 100 days. And that means that the T cell has to be able to do a very good job against the two molecules when they're together, but against each one alone. Then I want to discuss this if-better gate. And that gets a little more complicated and more interesting. So this is another strategy that we're developing for acute myeloid leukemia. And so we surveyed the expression of candidate targets for this disease. AGRE2, CLEC12A, CD123. There's a missing label here, it's CD33. And these dots here represent these axes, X and Y, are not the fluorescence intensity, but rather the median number of molecules on the surface of these primary samples. Okay, so there's a lot of work here in measuring each patient sample. And you see here CD123 and 33 are abundant. And you might go after those molecules, that's attractive. And in fact, there are trials today targeting CD123 or CD33. However, this is on the right here, normal bone marrow. And you can see that CD33 and 123 are also expressed on some normal cells, common myeloid progenitors, etc. And we were concerned that we couldn't target this while avoiding these. These are the normal cells, right? We thought we had a better chance with this combination of antigens here because you see AGRE2 is only found on early hematopoietic cells, but at a level of about 1,000 molecules or fewer. And it is well known there's AGRE2 in some myeloid cells and neutrophils. So we thought, could we target these cells? It's maybe not every patient, but many of the patients, while sparing these cells that we want to protect. And for that, we created a CAR specific to AGRE2, and the helper molecule to antigen B, if you recall, is CLEC12A. And this is not a CAR. It doesn't have the activation domain, so it enhances binding to the second antigen. So I'll show you the outcomes in mouse models. Here is the treatment of an AML placed in NSG mice. And this cell line, MOLM13, well established in the field, has about 10,000 molecules of AGRE2. And you see this clone places us here on the spectrum of all patients, right? So we put this in mice and we treat them with these CAR T cells. And actually the CAR T cells work very well. Actually very, very well, because they're actually quite low doses of T cells. Now, you might ask, did this second molecule, the antigen B, the helper target, if you like, matter? Well, let's knock it out. So we create this variant line. There's no more CLEC12A. So now we're here, right? This line is positioned here relative to all patients. And the CAR T cells still work very well. Okay, there was enough A, they didn't need B. So now let's reduce the amount of antigen A of AGRE2 here. Now there's 1,000 molecules on these clones. So now we are here. You see this little circle here. And now the CAR T cells fail. This was exactly what we were hoping to see, actually, and so they fail. And now we can ask the question of the if-better gate. Okay, there's 1,000 molecules of A, but now there's abundance of antigen B. And now what happens? Well, now the T cells work again. And that's if better. There's still not much A, but they work better because antigen B is there. This is what we call context-dependent antigen sensitivity. And we started a clinical trial now at Sloan Kettering. And it's a dose escalation trial here. And I can't comment on results yet. We have to wait a little bit. But I can show you one piece of this, which is asking really, well, does this if-better gate, is this just fancy mouse modeling and fancy words, or does it really work in patients as well? It works in mice. And so I'll show you the profile of the first patient we infused. So this is CLEC12A and AGRE2 in their bone marrow and peripheral blood prior to CAR T cell infusion. And I want to show you the levels of molecules that we measure in these patients. So you see there's about 3,000-4,000 up to 3,000-4,000 molecules on the surface of AGRE2. There's a lot of CLEC12A, over 100,000 molecules of these, and there's double positive cells. And here's the marrow 2 weeks, after infusion of these CAR T cells. And while there's still some AGRE2 cells, but with a lower level than before, it looks like we're sparing the AGRE2 low, which you see, and none of those really have CLEC12A, because if antigen B was there, probably they would be destroyed. And there are CLEC12A cells that are left, of course, but those are negative for AGRE2. So it really looks like the if-better measured in preclinical studies is carrying over to the clinical setting. And of course, we'll need many more patients and more time to see where this goes. I'm going to if I still have a few minutes, discuss a bit of T-cell persistence. Yes, I do? Okay. Two aspects of persistence, very different aspects. The first is, can we affect the persistence, of these functional T-cells through, again, engineering of the CAR itself? And this brings us back to the very beginning of this talk and this work, when we created these fusions combining CD28 and CD3 zeta signaling domains. Again, it's the clinic that told us that these cells are effective. Obviously, they're FDA approved now, but they're short-lived, a few weeks, and they don't persist as longer as the 4-1BB CARs that I mentioned. The 4-1BB CARs persist longer. They're not as good effectors. Their effector functions are less, but they last longer. And that's why both are useful, really, but with different properties. One that's a better effector that doesn't survive very long, and the other one that's a milder, if you like, T-cell that persists longer. And at first, we and probably everybody else thought, well, that's just a feature of CD28 and 4-1BB. But we went back to this design and thinking, well, maybe the signaling strength of this receptor is a bit too strong. It's a bit of an irony because it took us 10 years before, back in the 90s, to figure out that we needed to strengthen the signal, but maybe it was just a bit too much. So we engineered these variants in the zeta chain, and the zeta chain is very well known in mouse immunology. It's been extensively studied, and we could it has these three domains called ITAMs, and there's ways to inactivate them one by one. And so we created these versions where you see here 1XX has the proximal ITAM functional, the two others are deleted, and a whole bunch of versions. This was published too, long ago. And so here I'm just showing you an example of how we compare these CARs, introducing them in human peripheral blood T cells and infusing them to mice that bear, immunodeficient mice that bear CD19 positive acute lymphoblastic leukemia. So this is the conventional CAR. Again, this has been given to probably 20 or 30,000 patients, and here's put into these mice, but at a very low dose. So you see there's responders, some long-term, but they tend to relapse. We call this a stress test. It's just hard for the T cell to work at this point because it's a low dose. And so here are CARs, same dose, but they have only one functional ITAM. And you can right away see this 1XX is really much better. So the other ones only have one ITAM, but it's not the same one. So anyway, it doesn't matter which one it is. I won't say much about the mechanism. There's a whole paper about that. But the bottom line is that we at least propose that what this CAR design does is that it does confer good effector functions to the T cells. That's what these heat maps show. But the signaling activation is not so strong that it turns off the persistence mechanism, if you like, the self-renewal and memory signals in these T cells. So it's a good effector, but it can persist longer. And I'm going to talk about that a little bit more in a clinical trial. When you hear me again, I'm sorry, a bit later today. So I'll show you what we found in the clinic with that receptor. But I'll just show you here that two other groups asked us for these sequences, and they did a comparison that we had not done, comparing it to those classic, again, 28 zeta and 4-1BB zeta CARs in solid tumor models in ovarian cancer and breast cancer, targeting either mesothelin or this piece of a molecule called MAC1. And in both studies, ovarian and breast cancer, they found that the 1XX outperformed either one of those two classic variants. So needless to say, this 1XX CAR is what is the only CAR we intend to use going forward. Lastly, I want to comment on another aspect of the persistence of CAR T cells, very different, in the allogeneic setting, and let me explain what that is. So what are the sources for these T cells? Well, the field started with using autologous cells, the patient's own cells, of course. And they're engineered in a laboratory, ex vivo. Some people think that this engineering may disappear. I doubt it. It is making so much progress. It used to take 2 weeks, then 7 days. Now there are protocols that are 3 days. We're all getting better at that. So, and it is, I think, the method of choice, and I think it's here to stay. Having said that, it would be interesting to have cells ready to go that you don't have to make for every patient. You know, some people use the term off the shelf, right? So those would be allogeneic. They would be cells from someone else. And there are two main sources. You can collect them from selected healthy donors or create these T cells. This is something we're interested in that I'm not going to talk about today, where the T cell didn't come from a human. It wasn't collected from a human being. It was born in vitro in a bioreactor starting from pluripotent stem cells. So anyway, but in any case, these are allogeneic cells. If they're allogeneic, here you're going to run into this issue that your patient is going to reject those cells, right? So it's attractive to have off-the-shelf cells, but they might be rejected. And they might be rejected through antibody recognition, or the host T cells might reject these CAR T cells. And even if you ablated HLA class 1 on those CAR T cells, then the hosts, the recipients, the patient's NK cells would reject these CAR T cells. Nonetheless, it's a very interesting approach, a very interesting problem. And so the question is, can you prolong, if you like, the persistence of these cells in the recipient? They don't have to be permanently accepted. It's not a heart transplant or a kidney transplant, but they have to last long enough in the recipient that you could get a therapeutic response. So what I'll show you here is just two things. One, most importantly, is the model that we now established. This is going to be published, I think, in a week or so. And is the model that we established to study these allogeneic CAR T cells. So this is the immunodeficient mouse. The readout is, are the CAR T cells working? So they have this, the mice have this CD19 positive leukemia, and we treat them with CAR T cells from, let's say, donor A, here as usual. But before we give these CAR T cells, we fill up the mouse with either more peripheral blood mononuclear cells from the same donor, that's the autologous, or a different donor called the allogeneic, completely mismatched. And the important detail of the model, here's the ratio. There's 50 times more of these PBMCs than a CAR T cell, so we're not trying to make it easy for these CAR T cells. And below here, I show you that if you just do this and measure how many CAR T cells are there on day 10 in the bone marrow, well, with the autologous, you measure a certain number of these T cells, but they're gone if it's allogeneic in this model. So these CAR T cells are wiped out by day 10. So you have to do something, indeed, for these CAR T cells to be not rejected so fast. And so I'll just show you one sample here of what we can do with this model and where we're going with this. So this is this allogeneic model, and here we're infusing the unmodified CAR T cells in black, so the tumor progresses, right? They are rejected before they had time to eliminate the leukemia. If we knock out HLA class 1 on these CAR T cells, you see that the tumor progression is even faster. You made things worse. And we show here, I'm not showing you here, that's because the NK cells of the recipient are now rapidly eliminating these incoming allogeneic CAR T cells. But we found that if we reduce HLA class 1 to about 20% of baseline, then these cells persist much longer. And the way we do it is by using, in this case, a viral protein called NEF, coming from none less than HIV. Which downregulates HLA class 1 and allows these cells to survive for a longer time. And I just I'm going to wrap up in a minute. I just want to show you that these cells are eventually rejected after 3 weeks. But you see at 2 weeks, for example, when they express NEF, there's about 100-fold more than the natural types. So it's a very significant enhancement, if you like, of the persistence of these allogeneic CAR T cells in this pretty stringent model. And so I'm just going to finish here by saying, well, these were my illustrations of this explosion in our field now where, bioengineering is coming together with immunology to develop these new cell-based therapies. I think the reason we're attracted to these therapies is that they are at least sometimes curative. not just delaying progression of the disease. Of course, they're not curative for all, but that's why we need more research. And more importantly, they're given once. Okay, the living drug is, you give it once. And that, I think, is very attractive and explains, why so many people want to, make this now work even better. And so we have all these wonderful tools. There's all these new CAR designs. I tried to show you a few today, HIT and others. More and more cell types are investigated, not just peripheral blood T cells, or so-called alpha beta T cells, but gamma delta T cells, NK cells, natural killer T cells, macrophage, B cells, neutrophils are being engineered. Genome editing, I didn't show much of that, but is now a valuable addition to reprogram these T cells. I mentioned briefly the possibility that T cells might be generated from pluripotent stem cells. You could create immune responses in bioreactors, bypassing vaccines and even a normal human T cell donor. And then in vivo engineering is also just starting. Hard to say where that will go, but using either lipid nanoparticles, delivering mRNA, or we're working with this group, Laureate, we're using lentiviral vectors that are coated with polymers to retarget the delivery in vivo. And of course, where this leads us is new cell types, new engineering modalities, this really new applications. So you see the tip of the iceberg was the CD19 CAR therapy, but there's much more below the iceberg. Myeloma is starting to show interesting responses. Solid tumors, we know, are still a challenge, but I see no reason why they won't succeed. And autoimmunity is emerging today as a major focus for CAR T cells. That's something I'll discuss briefly in the second lecture. And so I don't have yet a slide from my new institution at Columbia, so this is largely my team as it was before I left MSK. There's members of my lab, of course, and we had established this manufacturing laboratory led by Isabelle Riviere, which is the critical tool needed to translate all of your wonderful ideas into first-in-human trials, and perhaps to even produce these medicines for patients beyond just the trials. Thank you for your attention.
A great deal of work has been done by you for the CAR T cell therapy, and there's a lot of interest with solid tumor. And I was just wondering whether you can share with us your perspective. Would the targeting and the delivery system be key in the solid tumor? And if so, and other things, what is your approach to that?
Yeah, thank you for the great question. You see the solid tumors here are a big piece of below the iceberg. So as you know, some investigators went to treat solid tumors with the CAR structures that were developed for leukemia and lymphoma. And the results are, not great. Not too surprising to me. Those CARs had been developed in hematological malignancies. And in those cancers, T cells traffic rather well into bone marrow or lymph nodes or spleen. The microenvironment of those tumors is not as, certainly not as structured as it is in solid tumors. And it's probably not as immunosuppressive as it is in most solid tumors. So I think it requires additional modifications to the T cells. I'm convinced that it will work for solid tumors. When you do checkpoint blockade, when people give these antibodies that unleash suppressed T cells, it's the T cells that do the job in checkpoint blockade, right? The antibody itself doesn't touch the tumor, it's the T cells that do the work. So we know very well that T cells can eliminate tumors, that there's no doubt about that, but they have to be engineered differently. So when I talk about sensitivity, when I talk about persistence, all of those are building blocks to tackle solid tumors. And then there's the one more that I did not talk about, which is the microenvironment. Different tumors, so most tumors create a microenvironment that protects the tumor, right? It feeds the tumor and protects the tumor from immune rejection. But a lot of those mechanisms have been identified. There may be some yet to be discovered, but a lot of them are known. The question is how to engineer the T cell now to overcome those mechanisms. And that's what you're seeing in the preclinical literature. There's a lot of that work right now. Is it, TGF-beta? Is it this, that? And so you're going to need T cells that are sensitive, that persist, and that can resist the microenvironment. And I believe that when that comes together, in the next 5 years, maybe 10 years, that we'll start seeing big successes in solid tumors.
Yeah, thank you very much. Excellent talk. Can you please comment on any research aimed at minimizing toxicity? As you know, from the clinician point of view, these treatments work very well, and clinicians are getting better at managing toxicity, but still on occasions, this kind of treatment can be fatal on rare occasions. Any progress on research aimed at minimizing toxicities of these cell therapies, please?
Yeah, another great question. And I did mention the toxicities, even as I said, I could talk about it for an hour. I just devoted one slide to that. It's very important to understand, the mechanisms of these toxicities so that we can treat them and ultimately prevent them, right? And it's because of these toxicities that the patients right now have to be admitted. If we had very few toxicities, we could do this as outpatient, and that would be very useful. Or at least the patient would have to be in the hospital even if they stayed nearby. But until then, it's a big imposition. Of course, there's a risk for the patient, and it's an imposition to have to hospitalize these patients when they receive the therapy. So the cytokine release syndrome is pretty well understood. In the very first trials, we didn't expect it. We had deaths on these trials at MSK, at Penn, at the National Cancer, everywhere. Just a few, but they were so unexpected. Nobody thought this would happen. The mouse models didn't show that. And this goes to a feature of these cell-based medicines. They're autologous, right? And every patient's T cells is different. And in some patients, there's an explosive response, a huge proliferation. And in others, not so much. In fact, if there's little proliferation, that's usually a failure. It's not too common. But in some patients, there's much more rapid expansion, and that tends to be the ones who develop more toxicities. As you know, the group at Penn identified IL-6 receptor blockade, and of course everybody uses corticosteroids. We established a mouse model, and then we found out that IL-1 receptor antagonist prevents this syndrome, but you have to give it preventively twice a day in these patients. Not everybody wants to do that, so there are ways to mitigate the cytokine release syndrome. So I think that today it's not lethal, but it requires careful monitoring and thoughtful and rapid intervention if that patient happens to be one or there's CRS developing. The neurotoxicity is a little bit more complicated. Nobody knows exactly how it works. With Mohammed Hamieh, whom you might have mentioned on the slide, we developed a model. We have a mechanism that we're looking at right now that we think accounts for the damage to the blood-brain barrier, but I think nobody really knows, and this neurotoxicity is an issue. Until we understand it better, it will be hard to understand. It doesn't require the entry of T cells in the brain. In fact, in glioblastoma, in brain tumors, you can put lots of CAR T cells in the brain, and you don't get this neurotoxicity. So there's something else coming from strong peripheral T cell activation that leads to some damage in the brain. We know there's microglial activation. There are cytokines produced in the central nervous system, but we don't know yet how well it works. So yes, there is research on these to try to understand them, and hopefully we'll reach the point where we can either, at the minimum, detect it early in the patient, so very early you know what to do, but ultimately prevent them. I'll mention something later about another CAR which, because it uses a much lower dose, gives very little of this neurotoxicity. So even if we don't understand it, being able to treat patients with lower doses seems to already greatly reduce these toxicities. Yes?
I have a quick question. I'm just going to go back to the solid tumors question. You know, it's a term that encompasses an extreme genetic heterogeneity. I'm a melanoma and sarcoma pathologist, so you know, UV-driven melanomas have a high TMB, and immunotherapy works pretty well. And I have the sarcoma with complex phenotypes. We don't understand them very well, but I'm more interested in the fusion-driven sarcomas who have a low TMB, immunotherapy doesn't work. Have they been tried in fusion-driven sarcomas, like synovial sarcomas, etc., and do they do anything?
No. We were going to start a collaboration with Bill at MSK, but we never went to sarcomas. That would be a very attractive cancer to tackle. Of course, we know how difficult it is to treat. There has been some T-cell receptors, you know, at the fusion. So the initial idea was that there are many fusions that underlie these different sarcomas. And the fusion points of these exons create peptides that could theoretically be targeted by a T-cell receptor. It goes back to my comments at the very beginning on the probability that a given patient will have a T-cell receptor for that fusion peptide. And so no, I think sarcomas would be a great place to look at and to explore, through proteomics and other ways to look for what's unique on their cell surface.
I think for the sake of time, we'll take the last question from there.
Okay.
Thank you for the outstanding talk, and Rich, of course, and I would like the chance to welcome you again, Saudi Arabia. My question is regarding your last image of Equium work, where you used site-directed insertion of CAR, where you delivered the template using the AAV and CRISPR. Do you think the field will be moving towards the site-directed insertion, where you showed more physiological expressive CAR T-cell, less exhaustion and less tonic signaling? And my second part of question regarding the FDA press announcement in 2023, do you think this could be linked to, again, the viral vector, the integrated viral vector delivery of the CAR, which we can see all the FDA approved now, they are all viral vector. Do you see the field that's moving toward non-viral and AAV delivery rather than using viral vector in the future?
It's a great question. I can't predict, but for everyone, it's a great question, very specialized question. The viral vectors that are used by most today randomly integrate in the genome. So, if you take 100 cells, the vector is integrated in 100 different locations in each cell. So every cell is a little different. And so, because depending on where the vector integrates, the chromosome influences how the transgene will function. So obviously it works. There's an average behavior, if you like, in these cells. But if you had precision engineering, you could make T cells that would be all very consistent. The FDA would tend to like something that's very homogeneous and consistent. The other features, as you mentioned, is that if you express this in a controlled level, at a right level, you can mitigate issues like tonic signaling, etc. So all those are attractive features. On the other hand, it's kind of expensive. And we have treated patients now with track engineered CAR T cells. I didn't bring slides on that. We treated 6 patients. It was a lot of work. The FDA approved it, although they asked for so many tests, and they made it so expensive that frankly, we don't know if we're going to continue that. Now, for some companies, maybe that's not a problem, but remember also they charge huge amounts, huge prices for their products. So I don't know where it will go, but it is certainly biologically very active. As you suggested too, there's maybe a better safety profile. Having said that, the lymphomas with these viral vectors are really rare. And we don't know yet if these genome-edited T cells will also not show some risk. We expect not, but none of these patients have been out 5 years or 10 years either, so we think it's safe, but we don't know. So anyway, I would tend to say that yes, it is a preferred direction going forward, but it's a qualified yes, I guess.
Thank you so much.
So these are a few additional updates to, I guess, what you already heard about earlier. Yeah, here are my disclosures, none of which are really relevant to this presentation. So you remember we talked about these chimeric antigen receptor molecules, and these are the first two that were approved by the FDA. And you recall that they are characterized by this dual signaling machinery that comprises an activation domain, typically borrowed from the CD3 zeta chain, together with a costimulatory domain, either borrowed from CD28 or 41BB. These two designs account for over 95% of the 1,500 or plus CAR T cell trials that are listed on clinicaltrials.gov. And you remember I started telling you that in the previous talk that we revisited this original design, and because the clinic had showed us that these CD28 based CARs are relatively short-lived, so while they're very good effectors, we would like them to persist longer, which I think would be a critical step to tackle solid tumors. And you may recall that I mentioned to you that we re-engineered the activation domain. The CD28 domain was unchanged, and that in fact by reducing, not increasing, but by reducing the number of these activation domains, we reached a better equilibrium between strength of activation, effector function, and persistence of the cells. And this is in animal models, right? So of course the question is, well, does the animal model finding hold up in clinical studies? So we took this to a clinical trial. So here's the CAR. It's a 19 but 1XX, right, with this attenuated activation domain. In this case, we changed the binding segment of the CAR, which is not relevant to what I will discuss, but I'll mention it. So it was changed. These were the two scFvs that are, I showed before. This one, FMC65, being the one that is so commonly used, and we identified another here only because it's human, it's not mouse. So anyway, but the key point here was to evaluate this 1XX domain. And so we decided to proceed in a trial with patients with diffuse large B-cell lymphoma. And the question is, well, how would you tell if is your CAR T cell better than others? You would formally need a side-by-side comparison, which I hope will eventually be done. But we thought we could already get an insight into that by designing the trial in the following way. And here I direct you to this yellow box here first. I want to start from the commercially available CAR T cells. And this one here, Tisagen, if you like, is the 41BB based CAR. It's actually sold by Novartis, and it's been used in a lot of patients. But note the dose. A typical dose in human patients is about 300 million CAR T cells. Here's another one called Axi or Axi-cell. This is a CD28 based CARs. This one is sold by Gilead now. And the dose is around 150 or so million CAR T cells. This product is a bit different because it's manufactured in a different way from the other two. So essentially doses of 1 to 300 million CAR T cells. So we thought, well, really, if this 1XX CAR T cell is, let's say, better than those, it should provide results, clinical outcomes that are at least as good as the other ones, but at a lower dose. So that was the way we thought we could start this trial. So it's a dose escalation study, and notice this starting dose is 25 million CAR T cells, right? So it's far fewer than those. And so we did the dose escalation, and here I'm just showing you the complete response rate at every dose level. So every cohort is small, right? It's 3 to 6 patients only. But you see the complete remission rate is on the order of 3/4, 75%, which is actually very good for a DLBCL, where with commercial products, it's less than that, although it's an unfair comparison because this is just a small study. But anyway, it is very good. And we didn't see a dose response, right? You see that? And so what we did not do what one usually does in a clinical trial, which is to do the expansion cohort at the maximum tolerated dose, but we thought it was more informative to do the expansion cohort at the lowest dose, since it seems to be as effective as any other one. And so we infused 12 more patients at the dose of 25 million CAR T cells. And here's the outcome, essentially, 75% complete remission rate, which is very good for this, diffuse large B-cell lymphoma. We show in the paper here that these patients are as severe as any other study, really. And secondly, we had predicted, and this goes to a question earlier about toxicities, that if you can treat patients with lower T-cell doses, you might encounter fewer toxicities. And that's indeed what you see. There's only one in these 16 patients that received 25 million CAR T cells. There's only one that has a significant cytokine release syndrome, and zero, not a single one of them has a neurotoxicity. So it does fit that a lower dose would be less toxic, but above all, we're getting these complete responses rates with these very low doses. Now, we waited for a long time to publish these results because the other question, before you ask me, would be, are these complete remission responses durable? Yeah, they are. So we waited for 2 years in this whole study before writing up the paper. And these are really as durable as any other CAR T cell response. So what it means is that you can achieve the same results as with, again, some of the well-established commercial products, but with a 10 times lower dose. So I think this supports this principle that if you keep improving on the design of the CAR, you can make better and better T cells, and the doses will go down, which means the manufacturing costs, the duration of culture, and perhaps for those who might go in vivo someday, that there are fewer T cells that you need to target. And I think the doses will probably keep going down even lower than 25 million. So these CD19 CAR T cells have been developed to tackle these B-cell malignancies, leukemias and lymphomas, and that's where these approved products, all exist today. But then something happened a few years ago. A group in Germany, Andreas Mackensen and George Schett, used these CAR T cells in patients with autoimmune disorders. And they designed, it wasn't even a trial, it was on a compassionate basis that they infused CD19 CAR T cells to some patients with refractory systemic lupus erythematosus, right? A systemic autoimmune disease. And these were patients with, that they called refractory to various immunosuppressive regimens and with progressing nephropathy, etc. And there was a first report of one patient, then there was this paper with 4 or 5 patients. And what they reported, and now has been observed at other centers, are complete remissions in patients with lupus following a single infusion of these CAR T cells. Now, these CAR T cells weren't tested, weren't developed for autoimmunity. In fact, one reason we had chosen CD19 some 25 years ago as a target was knowing that there would be a B-cell aplasia but that the B-cell aplasia in cancer patients would be acceptable, tolerable as a side effect, because it's better to be alive without B-cells than dead from, you know, your lymphoma or your leukemia. And now that side effect becomes the therapy, right? And so this has led, I call it a second, if you call the first, some people describe the CAR T cells as a tsunami in the world of medicine, and there's maybe a second one, and it's exactly the same CD19 CAR T cells. So this has expanded quickly beyond lupus, and you see this is from a review, a little commentary in Nature from last year, that some have started treating now patients with myasthenia gravis, or multiple sclerosis, or neuritis optica, or scleroderma with CD19 CAR T cells. And this is now kind of an explosion of trials, particularly in China and in the US. And this is just with one of my colleagues at Columbia here, Anka Askenazi. We're just reviewing the data that was presented at the big annual rheumatology meeting last fall in the US, where a whole bunch of these trials were presented. They're all early. Everything here is early. You know, you see the number of patients here, between 1 and 20 patients in these trials, so all these are starting. And I just want to, you don't need to read this, obviously. It just shows you there's a lot of activity suddenly in autoimmunity. And so most of these trials target CD19. A few of them BCMA, and one of them, the two antigens together. These products are available, right? So people started testing them. A lot of them use the 4-1BB CAR, but some also use the CD28-based CAR. Most of them are autologous, but a few are testing allogeneic cells. I mean, these products are available from cancer, and so they're going there. And so my point, though, about that is that it's wonderful. I'm thrilled that CD19 CAR T cells are finding more applications. But in autoimmunity, this developed, there was no preclinical research, right? All of this was done in oncology, and now it's available to autoimmunity. And I think that it hasn't been well appreciated that not all of these CARs are quite the same. I commented earlier on, the antigen sensitivity, the persistence, we know their toxicities, and you know the migration profiles of T cells are not the same either. Where do these T cells need to go in autoimmune patients? You know, is it in their lymph nodes, or is it in the central nervous system for some disorders? Is it in neurons? What organs? So one should not think that just any of these could work. And so I think that it's going to be certainly interesting to see what happens as these different trials report their data. I want to make the point that this 1XX CAR T cell that I just showed you, I think is very well suited because it's a low dose with very little toxicity, and that it too probably would fit well within these diseases. And then I just want to finish with, starting from autoimmunity, but I'll end on cancer again, is that, of course, there are many more targets being pursued in the field. For different cancers, people are searching for either single targets or combination of targets, targeting 2 or 3 or more antigens in solid tumors. But in the field of non-cancer, it's not just about CD19. There's another target I want to mention here called UPAR. And it stands for urokinase plasminogen activator receptor, and which is relevant to some autoimmune diseases, and call it senescence associated pathologies, where senescent cells accumulate in different tissues. And this work started with a collaboration with Scott Lowe at MSK and led by this very talented student, Corina Vegas. And we started doing proteomics on senescent tissues. Senescence was induced in different ways with oncogenes or drugs, and found that across these different senescence programs, this molecule, UPAR, was quite consistently upregulated. So it's on the cell surface, so it could be a CAR target. So this we published already a few years ago. And we showed that in different models in mice, we could induce liver fibrosis. We could do it with oncogenes, we could do it with a fat diet, we could do it with toxic compounds. And which showed the appearance here, you see the beta-gal staining in the liver of these different mice. And that we could then treat them with CAR T cells targeted to UPAR. And as you see, there's a disappearance of the senescent cells, and at least in these models, a reversion of the fibrosis in the liver. I'm not implying that would occur in disease models, but at least in this animal setting, the clearance of the senescent cells reverted the fibrosis in these models. So we're very interested in this, of course. And so all I want to show you is that, first of all, this target, UPAR, we see in a number of human pathologies now. And so here's our liver fibrosis here, and it's hard to see on this scale. But there's a number of these the yellow is hard to read, of UPAR cells, but you can see the increase scored by a pathologist actually at MSK, by Evelyn Filial here, that there is an increase of UPAR positive cells in human liver fibrosis, also in lung fibrosis. And so we think that, at least as a target, it is present in these tissues. And UPAR is upregulated in senescence, in normal cells, as well as in some cancer cells. You can actually induce senescence in cancer cells with radiation or some chemotherapeutic agents. And so we're also very interested in bringing UPAR CAR T cells to clinical trials. It might be more straightforward to do so in cancer patients than in patients with fibrosis. We haven't decided yet, but I just want to show you some. This is submitted right now, so this is not published either yet. This is ovarian cancer, and you see as ovarian cancer spreads, to the omentum, and you get this peritoneal disease. There's a lot of UPAR positive cells, and in fact, both the cancer cells. Ovarian cancer is one of the most commonly UPAR positive cancers, as well as some of the stromal cells. And so we now have these human binders for CARs that target UPAR, T1 and A1. And you see we induce this peritoneal carcinomatosis in these mice. You can see here with the firefly luciferase all this tumor in mice. The left here, this is patient samples on the left, this is human, but here on the right is mouse models. And you can see that these UPAR CAR T cells are actually very effective. So I think UPAR is a bit like CD19, another example of a target that's relevant to cancer, but also relevant to non-malignant diseases. And that would be great. Obviously that would accelerate the discovery and understanding of these CAR T cells. So anyway, I just want to go back to what I said before, which is, but I added something here, which is really not just the CARs, cell types, engineering modalities, etc., but new targets. And amongst these new applications, I think the one that is actually moving faster than the solid tumors, which I think will come, but will take more work, is really the application of these CAR T cells to some chronic disorders, and in particular autoimmunity. There are also trials that have recently opened in multiple sclerosis and some other disorders using C19 CAR T cells. So this is my conclusion here, is really there's so much more to do with T cell engineering, and I will stop here. Thank you.